Hello and welcome to your lecture on Chapter 19 on the factors affecting human performance. We are going to discuss the sites of fatigue, factors limiting all-out anaerobic performance, our factors limiting all-out aerobic performances, and the athlete as a machine. So we'll start off with this figure here showing us the factors that drive into performance. Every performance is going to require a certain amount of strength that we see and skill that we're going to need to be able to apply that strength by having practice and natural endowment. That is, of course, genetic, our body type, and our muscle fiber. Of course, we can train that aspect of it as well. We are also going to have to think about our energy production depending on what type is needed for the performance that we want. So are we going to use our anaerobic sources or our aerobic sources that we have extensively talked about in previous chapters? Then we can also consider our diet and our environment that are going to play a role, especially when it comes to endurance performance. And lastly, our central nervous system function, in which we are going to psychologically commit to go for the win or go for the gold, including arousal and motivation. So let's talk about our sites of fatigue. The word fatigue means that we have an inability to maintain that power output or force during repeated muscle contractions. And so we can have a central fatigue that involves our central nervous system or the brain and spinal cord, and a peripheral fatigue in which we're taking into account the neural factors, mechanical factors, our energetics for contraction, anaerobic or aerobic capacities that we were talking about. And then also we need to account for the uncertainty about the exact causes of the fatigue because we have found differences within research methods. So continuing our possible sites of fatigue, we are going to classify it as central, like we said, the brain and spinal cord, or peripheral, meaning outside of the brain and spinal cord. And you could see that depicted in this figure over here, where we see that central fatigue is going to take into account our psyche and our brain and information traveling into our spinal cord. So of course, as we move and transition that information, we have the idea of motivation or arousal that can send signals down and move into our motor unit recruitment. So this really is going to be the mechanism that shows our will to win. And then we could see it, like I said, moving down into the areas of our motor unit and then into the individual muscle fiber where we have the interaction of the contractile myofilaments that we've talked about. So this figure is really showing us the 9 points of the potential sites that are most recognized by our physiologist. And so let's walk through it really quick. The peripheral nerve is basically where we leave the spinal cord and send that nerve over to our muscle sarcolemma. That was, again, allowing the neurotransmitter acetylcholine to move across the synaptic cleft and on to the muscle acetylcholine receptors, allowing sodium to move into the cell and making it the inside more positive, triggering an action potential. And that is going to open up our calcium stores within the sarcoplasmic reticulum, and that calcium then goes and binds to troponin affiliated with the actin myofilaments, allowing the tropomyosin to move out of the way, and the myosin heads to bind to actin, causing a cross bridge, building tension and heat release as a result, and then the force being produced. Now next we have this table that discusses the advantages and disadvantages of various approaches to the study of fatigue. So first, let's talk through examples of experimental approaches that may provide a different scientific interpretation for what the cause of fatigue is. So first we have our muscle fiber type and training state. Next, we have whether the muscle contraction was initiated voluntarily or was it stimulated electrically? Third, the use of experimental muscle preparations, whether we are using a isolated fiber, which could be from an amphibian or a mammalian host. And then we've got our intensity of and duration of the exercise, whether that exercise was continuous or intermittent. So looking at the muscle in vivo, we can talk about the advantages here. Obviously, all of our physiological mechanisms are present, and the fatigue could be central or peripheral since we've got both systems in place. All types of fatigue can be studied, and the stimulation patterns are appropriate for the fiber types and the stage of fatigue. The disadvantage is each person is going to have a different mixture of the fiber types. We have complex activation patterns and lots of components there, and it produces correlative data that is hard to identify the specific mechanisms that take place. And experimental interventions are going to be limited here. Then if we take a look at isolated muscle, the advantage is that we don't need to really talk about the central fatigue, so we can focus on peripheral fatigue, and the dissection is simple. The disadvantage is we're still going to have that mixture of fibers here, and it is inevitable that we have extracellular gradients of oxygen, carbon dioxide, potassium, and lactic acid. Our mechanisms of fatigue are going to be biased by the presence of extracellular gradients, and then our drugs can not be applied rapidly because there is a diffusion gradient within the muscle cell. Next is going to be the isolated single fiber. The advantage here is that only one fiber is going to be present, so we don't have to worry about the mix, and our force and other changes are going to be ionic or metabolic that could be unequivocally correlated. We also have fluorescent measurements of the ions, our metabolites, membrane potentials, and lastly, it's easy, and we've got rapid application of the extracellular drugs, our ions, and metabolites. The disadvantages are that the environment is different to an in vivo, meaning in the actual body type of circumstance. Our potassium accumulation and other in vivo changes are absent at this point. It's prone to damage because of the temperatures, and we have a small size that makes the analysis of metabolites more difficult. Lastly, we have a skinned fiber. The advantage is that we have precise solutions that can be applied. We have possible to study myofibril properties, our sarcoplasmic reticulum, release and uptake of that calcium, and the action potential that is associated with that. And then we've got our metabolic and ionic changes associated with fatigue that could be studied in isolation. The disadvantage is that we don't have the relevance to fatigue, and we may lose important intracellular constituents, and the relevant metabolites to study have to be identified in other systems. So why do we think central fatigue takes place? It's that those motor units are activated and the motor unit firing frequency has increased. And what is unique here about our central nervous system is that we can alter the state of fatigue by facilitating that motor unit recruitment. And we could do this by increasing our motivation, whether it's external or internal, and also having that physical or mental diversion. The excessive endurance training or overtraining could cause central fatigue. We can have reduced performance and prolonged fatigue, and that is related to the neurotransmitter serotonin within the brain. And what we see evidence of is that when we exercise, we increase the amount of serotonin within our brain, so that as we have prolonged exercise, that can tend to decrease or delay the fatigue responses. There's also another study that indicates that fatigue isn't due to serotonin levels alone, but it's actually taking a look at the ratio of serotonin to another neurotransmitter known as dopamine. So on one hand, when we have a high serotonin and dopamine ratio, that can contribute to tiredness, while when we have a low ratio, that is linked to arousal and a better performance without fatigue setting on too quickly. And we've also found that low levels of norepinephrine can also contribute to fatigue. And we've talked about in past chapters how we have a central governor model in which we have our conscious and subconscious brain involved more with the fatigue levels and not really considering the spinal cord or the motor unit. But now we can, as we kind of stop talking about the central fatigue, we can generally agree that the central nervous system is going to contribute to about 10 to 15% of fatigue being onset compared to peripheral factors. So now we are going to talk about more of peripheral fatigue. This is relatively a minor contribution of the CNS to fatigue. The vast majority of evidence is pointing to various sites along the periphery here, where we're going to talk about neural factors first, and then our mechanical factors and energetic events that will kind of hamper the tension development within the muscle itself. So with our neural factors, we're seeing that the neuromuscular junction is not going to be an area where we have fatigue develop. We know that as that action potential transitions from the neuron over to the sarcolemma of the muscle, it is going to travel along the sarcolemma and enter the transverse tubules, also known as our T-tubules, and that will then allow our calcium to exit the sarcoplasmic reticulum, and it makes its way over to our troponin on our actin filaments. Now note that as this happens, we are going to have an altered muscle membrane to allow the conduction of those action potentials. And our sodium potassium pumps at that point are unable to reset the resting membrane potential until that action potential fully moves through. And so this can be improved with training, but sometimes what we can see that contributes to fatigue is that that action potential can be blocked within the T-tubules. And if it's blocked, that doesn't allow the calcium to be released from the sarcoplasmic reticulum. So when we have a reduction in that, we will have less calcium released, which reduces, of course, the force production of the muscle. Now let's talk about mechanical factors. We know that when that myosin head binds to the actin filament, we get that cross-bridge cycling that takes place and tension develops. So it is going to depend on the arrangement of the actin and myosin filaments and that ability of the calcium to bind to troponin in order to shift the tropomyosin out of the way. And we also know that we need ATP present so that myosin head can release the actin filament and then reattach. We have talked about how a high hydrogen ion concentration may contribute to fatigue, and this is because we could see a reduction in the force per cross bridge that forms. The reduction of that force generation at a given calcium concentration will also contribute to fatigue. This is because we have that relation between the hydrogen ions interface with the calcium binding to, that should say troponin. I don't know how that extra T got in there. But basically, all in all, we are going to have an inhibition of that calcium release from the SR, reducing the cross-bridge cycling. We can also have a longer relaxation time of that muscle. That could be a sign of fatigue, and that's because the cross-bridge cycling could be taking place much slower than originally. Then we have our energetics of contraction. This takes place when we have an imbalance between our ATP requirements and the ATP generating capacity. So this can happen when we have a high accumulation of inorganic phosphate that will then inhibit the maximal force, and we aren't going to be able to form as many cross bridges with our actin, because if you remember, we need ATP present for that myosin head to release from actin. And we'll also see that accumulation of inorganic phosphate can inhibit calcium release from the SR, sarcoplasmic reticulum, which minimizes the ability of troponin to shift tropomyosin out of the way for the myosin heads to attach. The rate of ATP utilization can be slowed faster than the rate of ATP generation. So basically, this is an issue of supply and demand. The demand is high and the supply is not keeping up. And so we need to try to maintain our ATP concentration and the cell not to run out of its ATP so we don't have fatigue take place. Another factor is going to be our muscle fiber recruitment increases as the exercise intensity increases. So you can see that as that exercise intensity is starting to increase, we are going to activate more of our muscle fibers. So first you could see we have our type 1 muscle fibers become recruited, and then we move into the type 2A, and lastly our type 2X. And you could see from this graph that up to 40% VO2 max type 1 fibers are recruited. This is also known as our slow twitch muscle fibers. And then our type 2A fibers are recruited at 40 to about 75% of VO2 max. And then we've got our exercises that are higher than 75% VO2 max is going to require our type 2X fibers. These are going to be our anaerobic glycolytic fibers, and that results in an increased lactate and hydrogen ion production. Another contribution to fatigue we've talked about is that free radical production is going to start to increase, and then that leads to muscle fatigue during our prolonged exercises. So those free radicals are going to be molecules with their unpaired outer orbital electron. And like we said, those electrons like to be stable. And so what it does is it looks for other molecules to bind with. And so it is capable of damaging our proteins and lipids within the muscle. This can contribute to fatigue during exercises that are greater than 30 minutes and can cause damage to the contractile proteins, so myosin and troponin, and can limit those cross bridges that take place between myosin and actin and creating that strong binding state. It can also depress the sodium-potassium pump activity, which could then disrupt the potassium homeostasis. As we know, when our muscle is at rest, we want lots of potassium inside and our sodium to be outside of the cell. And when we get or are moving toward an action potential taking place, we move more sodium in. And as the muscle then recovers from that contraction, we want to be able to move the potassium out of the cell. We've talked about in the past how antioxidants are able to reduce the amount of free radicals in our body, but we have seen that antioxidant supplements unfortunately do not prevent fatigue in general. When we do have a high antioxidant dose, it can impair the muscle's performance, and only N-acetylcysteine has been shown to delay exercise-induced muscle fatigue. So here on the right-hand side, we could see figure 19.3, and this is showing us a rested muscle. And here at the letter A, we are seeing that it is, the rested muscle is in its reduced state. So at the start of exercise, when we are producing force, we are going to see that there's a beneficial effect on the muscle force generation, as the initial production of free radicals is going to actually improve performance without muscle fatigue taking place. So as we approach B here, we are increasing the duration of our exercise and starting to go over 30 minutes. And that is going to result in a continuous production of the radicals, which eventually is going to cause a detrimental effect. And so as we continue that exercise, we are going to enter recovery, and fatigue will eventually start to dissipate as the redox state of the muscle is becoming more reduced through our full transition and back to our rested state. So next we're going to talk about the factors limiting all-out anaerobic performances. We mentioned earlier that as exercise intensity increases, we'll start to change our muscle fiber recruitment from starting at type 1, which is our slow twitch muscle fibers, into type 2A, which is going to be kind of like that mix of aerobic and anaerobic components of the muscle fiber, and then into type 2X, which is going to be those anaerobic, glycolytic, energic systems. And so what we see is as we start transitioning, our ATP supply that we need to create tension within our muscle is going to be dependent on anaerobic metabolism. So fatigue is going to be specific to the nature of whatever exercise task we are trying to perform, and the fiber types we'll utilize is specific to that task. So our anaerobic performance are examined scientifically based on exercise duration. So if we have an ultra short-term performance, that means it's 10 seconds or less, or short-term performance is between 10 and 180 seconds. So first, let's take a look at that ultra short-term performance. Again, these are events that are less than 10 seconds, so we call them high-power events, and we are going to need to recruit the type 2 muscle fibers, or fast-twitch muscle fibers, that are able to generate great forces in a short amount of time. And so we are going to need to utilize our motivation within our CNS, our skill, and arousal. The primary energy source is going to be anaerobic, as we said before. So we will really use that ATP-PC system in which we create one ATP from breaking off the phosphate from creatine, and then our glycolysis in which we use our glucose molecules and can generate 2 ATP per glucose molecule. And so we can see that within this ATP-PC system that we might be able to use creatine supplementation in order to improve the performance. You could see here that we've got practice and our skill and technique that comes into this type of performance, as well as our CNS motivation and arousal, choosing the right fiber distribution and recruitment, and using our energetic systems in order to create muscle power to increase performance. Now for our short-term performances, these are events that last between 10 and 180 seconds, and we can shift from anaerobic to aerobic metabolism. We'll see that 70% of the energy supplied anaerobically is at 10 seconds, and then we make a shift to 60% that will be supplied aerobically at 180 seconds. And in this figure down here, we can kind of see the distribution of that, where between 10 and 60 seconds, we are going to use predominantly our fast twitch muscle fibers. So you can see where our oxygen is being taken into account, our aerobic processes versus our anaerobic processes. And in that, we will see a result of elevated lactate and our hydrogen ion levels, and these can start to interfere with our calcium binding to troponin, which minimizes the amount of cross bridges that can take place, and will interfere with the glycolytic ATP production, which also would do the same. And so we can have a ingestion of buffers that can possibly improve performance. Now let's talk about the factors that limit all-out aerobic performances. So as we increase that duration of our performance, we will have more demand on our aerobic sources for energy. So there are some environmental factors that are brought into this, such as heat and humidity. We also can factor in our diet and hydration. That plays an essential role. So the way that aerobic performances were examined were based on these three types of performances. We've got our moderate length performance, which is from 3 to 20 minutes, our intermediate length performance, which is 21 to 60 minutes, and our long-term performances, which are 1 to 4 hours. So we'll start exploring the moderate duration performances. Like we said, that's between 3 and 20 minutes. And what we could see is that 60% of ATP is generated aerobically at the 3 minutes, whereas as that exercise continues closer to 20 minutes, we see that 90% of the ATP is supplied anaerobically. And so a high VO2 max is going to be an advantage at this point. It means that we have high maximal stroke volume. Remember, that's the amount of blood that we are pumping out of the left ventricle, and a high arterial oxygen content is going to be helpful in getting a higher VO2 max. So making sure we have a lot of hemoglobin that can bind the oxygen, and of course being able to inspire that oxygen to bind to the hemoglobin. And you could see that kind of written out over here, showing that the oxygen has to bind in order to contribute to the VO2 max. And then we are going to require energy expenditure near VO2 max. So our type 2 X fibers are recruited, and we have a high level of lactate and our hydrogen ion accumulation. So let's take a look at the figure on the right one more time. We talked about having that high stroke volume, and this is based on your genetics and your training. And if you remember, our stroke volume multiplied by our heart rate resulted in our cardiac output. So we want a high cardiac output to be able to contribute to our VO2 max. And we have this CaO2 representing our arterial oxygen content, and that is going to be influenced by our hemoglobin content and our ability to have that fraction of inspired oxygen and the amount of oxygen that is in the air. So this, of course, is going to be different if we are at higher altitudes. And then here we are going to have our fiber type come into play. It is based on genetics, what we were born with, more slow twitch or fast twitch. And then, of course, our mitochondrial and capillary density, which could be affected through endurance training. And so all of that is going to contribute to our VO2 max. Now let's talk about intermediate duration performances. These are events that are lasting between 21 and 60 minutes. So we will see that we predominantly will have a contribution through our aerobic processes, usually conducted at less than 90% of our VO2 max. And we want another high VO2 max here. Other factors are going to be our running economy and exercise efficiency, and this is going to be due to our biomechanical and other bioenergetic factors. In this case, measurements of both of our VO2 max and the running economy is going to be needed to predict how the performance will be. You could see that over on the right-hand side here. And of course, our percentage of type 1 muscle fibers will contribute to that. If we have more type 1 fibers, that will enhance the performance. And then we have to factor in our environmental factors. So heat and humidity will affect our percent VO2 max, our state of hydration, and also our lactate threshold. Now, what happens if we do have a high heat load? That's going to require for our cardiac output to be directed to our skin, pushing our cardiovascular system closer to maximum at any of our running speeds. So of course we want to reduce that in order to gain a higher aerobic performance. Next, we have our long-term performances. These are events lasting between 1 and 4 hours. These definitely will be through our aerobic processes, and we are going to have our environmental factors have a more important role in this type of performance. We'll maintain our rate of carbohydrate metabolism, which you can see that we are utilizing our liver and muscle glycogen stores, and we can supplement while we are exercising to assist with this and also change our diet in order to maintain our carbohydrate oxidation by the muscle. And we also want to consume more fluids and electrolytes in order to increase our VO2, and we have talked about our running economy already. That plays a factor here in our long-term performances as well due to the biomechanics and bioenergetics. So a question that was asked is maximal oxygen uptake important for distance running performance? And what they found is that VO2 max sets an upper limit for ATP production in endurance events. And so what this is stating is that the rate of ATP generation is going to be dependent on the actual VO2 that could be maintained during someone's run. So if we are not running at 100% VO2 max, we could see that a 2-hour and 15-minute marathon would require us to sustain a VO2 of 60 milliliters per kilogram per minute. And if we change and make our VO2 max at 80%, that's going to require a VO2 max of 75 milliliters per kilogram per minute. And so the VO2 max is really setting the upper limit for energy production in endurance events, but isn't going to necessarily determine the final performance. And so the performance is really dependent on that percentage of VO2 max at which that runner can maintain their performance, which is estimated through their lactate threshold and their running economy, which is going to have a dramatic impact on the speed that they can maintain over a distance. Next, let's take a look at this closer look on the factors that are going to affect performance in our ultra endurance events. So an example of an ultra endurance event would be a 166-kilometer mountain run, a triple iron triathlon, or a 24-hour run. So the most important variables for this is going to be the VO2 max and the percent of the VO2 max that could be sustained through the duration of the event. And our metabolic responses are fat oxidation, that is 3.5 times higher after the event, and it'll be consistent with exercise at less than 60% VO2 max. And we will have a response of 50% reduction in our muscle glycogen stores. This could be dangerous because we have a potential for hyponatremia, but it only tends to affect 4% of athletes. Now let's talk about the ability to break the 2-hour marathon. We've seen recent improvements in the marathon world record that have outpaced other endurance running events. Currently, there's an arbitrary goal that is breaking a 2-hour marathon. And so there was a study done in which they used 13 elite marathon runners, and they noticed that they possess a percent VO2 max at which the runner can maintain performance at 4 liters per minute, and that equated to about 13.1 or more miles per hour. And they were sustaining it approximately 67 milliliters per kilogram per minute. Their VO2 peak was approximately 85 milliliters per kilogram per minute, but this estimate was influenced by their running economy, and we know that that can also factor into it the biomechanics and also the bioenergetics within their body. So another factor would be their genetics and intense training that were both needed. So based on this scientific research, a unsanctioned sub 2-hour marathon occurred in 2019, in which 26.2 miles was run in an hour and 59 minutes and 40 seconds. And that is represented by this red dot over here, and raise the bar for all other endurance athletes. The last topic we're going to discuss is the athlete as a machine. And so we need to look at this as if we are trying to continue the goal of improving their performance rather than treating them like machines. And so the question is, are we trying to spot weaknesses that compromise performance and then recommend solutions? And so some people would say, yes, we are, and so this is a good thing, but others would argue that we are dehumanizing the athlete. So through our research, we are trying to understand how we're functioning and develop healthy and safe methods to allow the athlete to overcome personal limitations. But we also know that there are some athletes that are used by certain countries in order to advance their particular political doctrine, and the complicity of scientists who were recruited to make that athlete be able to run faster and longer and harder and jump higher. And so luckily there are these institutional review boards that are protecting the rights of the study participant, the athlete, and can help to minimize the risk of the subject being studied. And so that is it for chapter 19. We talked about the sites of fatigue and some factors that are limiting all-out anaerobic performance and aerobic performance, and the athlete as a machine.